Early plant life. The kingdom Plantae constitutes large and varied groups of organisms. There are more than 300,000 species of catalogued plants. Of these, more than 260,000 are seed plants. Mosses, ferns, conifers, and flowering plants are all members of the plant kingdom. Most biologists also consider green algae to be plants, although others exclude all algae from the plant kingdom. The reason for this disagreement stems from the fact that only green algae, the charophytes, share common characteristics with land plants, such as using chlorophyll, A, and B plus carotene in the same proportion as plants. These characteristics are absent in other types of algae. Evolution connection. Algae and evolutionary paths to photosynthesis. Some scientists consider all algae to be plants, while others assert that only the charophytes belong in the kingdom Plantae. These divergent opinions are related to the different evolutionary paths to photosynthesis selected. For in different types of algae, while all algae are photosynthetic, that is, they contain some form of a chloroplast, they didn't all become photosynthetic via the same path. The ancestors to the green algae became photosynthetic by endosymbiosing a green photosynthetic bacterium about 1.65 billion years ago. That algal line evolved into the charophytes, and eventually into the modern mosses, ferns, gymnosperms, and angiosperms. Their evolutionary trajectory was relatively straight and monophyletic. In contrast, the other algae, red, brown, golden, stramenopiles, and so on, all became photosynthetic by secondary, or even tertiary, endosymbiotic events. That is, they endosymbiosed cells that had already endosymbiosed a cyanobacterium. These latecomers to photosynthesis are parallels to the charophytes in terms of autotrophy, but they did not expand to the same extent as the charophytes, nor did they colonize the land. The different views on whether all algae are Plantae arise from how these evolutionary paths are viewed. Scientists who solely track evolutionary straight lines, that is, monophyla, consider only the charophytes as plants. To biologists who cast a broad net over living things that share a common characteristic, in this case, photosynthetic eukaryotes, all algae are plants. Plant adaptations to life on land. As organisms adapted to life on land, they had to contend with several challenges in the terrestrial environment. Water has been described as the stuff of life. The cell's interior is a watery soup. In this medium, most small molecules dissolve and diffuse, and the majority of the chemical reactions of metabolism take place. Desiccation, or drying out, is a constant danger for an organism exposed to air, even when parts of a plant are close to a source of water. The aerial structures are likely to dry out. Water also provides buoyancy to organisms. On land, plants need to develop structural support in a medium that does not give the same lift as water. The organism is also subject to bombardment by mutagenic radiation, because air does not filter out ultraviolet rays of sunlight. Additionally, the male gametes must reach the female gametes using new strategies, because swimming is no longer possible. Therefore, both gametes and zygotes must be protected from desiccation. The successful land plants develop strategies to deal with all of these challenges. Not all adaptations appeared at once. Some species never moved very far from the aquatic environment, whereas others went on to conquer the driest environments on Earth. To balance these survival challenges, life on land offers several advantages. First, sunlight is abundant. Water acts as a filter, altering the spectral quality of light absorbed by the photosynthetic pigment chlorophyll. Second, carbon dioxide is more readily available in air than in water, since it diffuses faster in air. Third, land plants evolved before land animals. Therefore, until dry land was colonized by animals, No predators threatened plant life. This situation changed as animals emerged from the water and fed on the abundant sources of nutrients in the established flora. In turn, plants develop strategies to deter predation, from spines and thorns to toxic chemicals. Early land plants, like the early land animals, did not live very far from an abundant source of water and developed survival strategies to combat dryness. One of these strategies is called tolerance. Many mosses, for example, can dry out to a brown and brittle mat, but as soon as rain or a flood makes water available, mosses will absorb it and are restored to their healthy green appearance. Another strategy is to colonize environments with high humidity, where droughts are uncommon. Ferns, which are considered an early lineage of plants, thrive in damp and cool places such as the understory of temperate forests. Later, plants moved away from moist or aquatic environments using resistance to desiccation rather than tolerance. These plants, like cacti, minimize the loss of water to such an extent they can survive in extremely dry environments. The most successful adaptation solution was the development of new structures that gave plants the advantage when colonizing new and dry environments. 4 major adaptations are found in all terrestrial plants. The alternation of generations, a sporangium in which the spores are formed, a gametangium that produces haploid cells, an apical meristem tissue in roots and shoots. The evolution of a waxy cuticle and a cell wall with lignin also contributed to the success of land plants. These adaptations are noticeably lacking in the closely related green algae, another reason for the debate over their placement in the plant kingdom. Alternation of generations. Alternation of generations describes a life cycle in which an organism has both haploid and diploid multicellular stages. Figure 1. Haplontic refers to a life cycle in which there is a dominant haploid stage, and diplontic refers to a life cycle in which the diploid is the dominant life stage. Humans are diplontic. Most plants exhibit alternation of generations, which is described as haplodiplodontic. The haploid multicellular form, known as a gametophyte, is followed in the development sequence by a multicellular diploid organism, the sporophyte. The gametophyte gives rise to the gametes, reproductive cells, by mitosis. This can be the most obvious phase of the life cycle of the plant, as in the mosses, or it can occur in a microscopic structure, such as a pollen grain, in the higher plants, a common collective term for the vascular plants. The sporophyte stage is barely noticeable in lower plants, the collective term for the plant groups of mosses, liverworts, and lichens. Towering trees are the diplontic phase in the life cycles of plants such as sequoias and pines. Protection of the embryo is a major requirement for land plants. The vulnerable embryo must be sheltered from desiccation and other environmental hazards. In both seedless and seed plants, the female gametophyte provides protection and nutrients to the embryo as it develops into the new generation of sporophyte. This distinguishing feature of land plants gave the group its alternate name of embryophytes. Sporangia in seedless plants. The sporophyte of seedless plants is diploid and results from syngamy, fusion, of 2 gametes. The sporophyte bears the sporangia, singular, sporangium, organs that first appeared in the land plants. The term sporangia literally means spore in a vessel, as it is a reproductive sac that contains spores. Figure 2. Inside the multicellular sporangia, the diploid, sporocytes, or mother cells, produce haploid spores by meiosis, where the 2 in chromosome number is reduced to 1 n. Note that many plant sporophytes are polyploid. For example, durum wheat is tetraploid, bread wheat is hexaploid, and some ferns are 1000-ploid. The spores are later released by the sporangia and disperse in the environment. Two different types of spores are produced in land plants, resulting in the separation of sexes at different points in the life cycle. Seedless non-vascular plants produce only one kind of spore and are called hemosporous. The gametophyte phase is dominant in these plants. After germinating from a spore, the resulting gametophyte produces both male and female gametangia, usually on the same individual. In contrast, heterosporous plants produce two morphologically different types of spores. The male spores are called microspores because of their smaller size, and develop into the male gametophyte. The comparatively larger megaspores develop into the female gametophyte. Heterospory is observed in a few seedless vascular plants and in all seed plants. When the haploid spore germinates in a hospitable environment, it generates a multicellular gametophyte by mitosis. The gametophyte supports the zygote formed from the fusion of gametes and the resulting young. Sporophyte. Vegetative form. The cycle then begins anew. The spores of seedless plants are surrounded by thick cell walls containing a tough polymer known as sporopollenin. This complex substance is characterized by long chains of organic molecules related to fatty acids and carotenoids, hence the yellow color of most pollen. Sporopollenin is unusually resistant to chemical and biological degradation. In seed plants, which use pollen to transfer the male sperm to the female egg, the toughness of sporopollenin explains the existence of well-preserved pollen fossils. Sporopollenin was once thought to be an innovation of land plants. However, the green algae, coleochaetes, forms spores that contain sporopollenin. Gametangia in seedless plants. Gametangia, singular. Gametangium, are structures observed on multicellular haploid gametophytes. In the gametangia, precursor cells give rise to gametes by mitosis. The male gametangium, antheridium, releases sperm. Many seedless plants produce sperm equipped with flagella that enable them to swim in a moist environment. To the archegonia, the female gametangium, the embryo develops inside the archegonium as the sporophyte. Gametangia are prominent in seedless plants, but are very rarely found in seed plants. Apical meristems. Shoots and roots of plants increase in length through rapid cell division in a tissue called the apical meristem, which is a small zone of cells found at the shoot tip or root tip. Figure 3. The apical meristem is made of indifferentiated cells that continue to proliferate throughout the life of the plant. Meristematic cells give rise to all the specialized tissues of the organism. Elongation of the shoots and roots allows a plant to access additional space and resources. Light in the case of the shoot, and water and minerals in the case of roots. A separate meristem, called the lateral meristem, produces cells that increase the diameter of tree trunks. Additional land plant adaptations. As plants adapted to dry land and became independent from the constant presence of water in damp habitats, new organs and structures made their appearance. Early land plants did not grow more than a few inches off the ground, competing for light on these low mats. By developing a shoot and growing taller, individual plants captured more light. Because air offers substantially less support than water, land plants incorporated more rigid molecules in their stems, and later, tree trunks. In small plants such as single-celled algae, simple diffusion suffices to distribute water and nutrients throughout the organism. However, for plants to evolve larger forms, the evolution of vascular tissue for the distribution of water and solutes was a prerequisite. The vascular system contains xylem and phloem tissues. Xylem conducts water and minerals absorbed from the soil up to the shoot, while phloem transports food derived from photosynthesis throughout the entire plant. A root system evolved to take up water and minerals from the soil, and to anchor the increasingly taller shoot in the soil. In land plants, a waxy, waterproof cover called a cuticle protects the leaves and stems from desiccation. However, the cuticle also prevents intake of carbon dioxide needed for the synthesis of carbohydrates through photosynthesis. To overcome this, stomata or pores that open and close to regulate traffic of gases and water vapor appeared in plants as they moved away from moist environments into drier habitats. Water filters ultraviolet B, UVB, light, which is harmful to all organisms, especially those that must absorb light to survive. This filtering does not occur for land plants. This presented an additional challenge to land colonization, which was met by the evolution of biosynthetic pathways for the synthesis of protective flavonoids and other compounds, pigments that absorb UV wavelengths of light and protect the aerial parts of plants from photodynamic damage. Plants cannot avoid being eaten by animals. Instead, they synthesize a large range of poisonous secondary metabolites, complex organic molecules such as alkaloids, whose noxious smells and unpleasant taste deter animals. These toxic compounds can also cause severe diseases and even death, thus discouraging predation. Humans have used many of these compounds for centuries as drugs, medications, or spices. In contrast, as plants co-evolved with animals, the development of sweet and nutritious metabolites lured animals into providing valuable assistance in dispersing pollen grains, fruit, or seeds. Plants have been enlisting animals to be their helpers in this way for hundreds of millions of years. Evolution of land plants. No discussion of the evolution of plants on land can be undertaken without a brief review of the timeline of the geological eras. The early era, known as the Paleozoic, is divided into 6 periods. It starts with the Cambrian period, followed by the Ordovician, Silurian, Devonian, Carboniferous, and Permian. The major event to mark the Ordovician, more than 500 million years ago, was the colonization of land by the ancestors of modern land plants. Fossilized cells, cuticles, and spores of early land plants have been dated as far back as the Ordovician period in the early Paleozoic era. The oldest known vascular plants have been identified in deposits from the Devonian. One of the richest sources of information is the Rhynie Chert, a sedimentary rock deposit found in Rhynie, Scotland. Figure 4, where embedded fossils of some of the earliest vascular plants have been identified. Paleobotanists distinguish between extinct species as fossils and extant species, which are still living. The extinct vascular plants, classified as zoosterophylls and trimerophytes, most probably lacked true leaves and roots and formed low vegetation mats similar in size to modern-day mosses. Although some trimetophytes could reach 1 meter in height, the later genus Cooksonia, which flourished during the Silurian, has been extensively studied from well-preserved examples. Imprints of Cooksonia show slender branching stems ending in what appear to be sporangia. From the recovered specimens, it is not possible to establish for certain whether Cooksonia possessed vascular tissues. Fossils indicate that by the end of the Devonian period, ferns, horsetails, and seed plants populated the landscape, giving rising to trees and forests. This luxuriant vegetation helped enrich the atmosphere in oxygen, making it easier for air-breathing animals to colonize dry land. Plants also established early symbiotic relationships with fungi, creating mycorrhizae, a relationship in which the fungal network of filaments increases the efficiency of the plant root system, and the plants provide the fungi with byproducts of photosynthesis. Career connection. Paleobotanist. How organisms acquired traits that allow them to colonize new environments and how the contemporary ecosystem is shaped are fundamental questions of evolution. Paleobotany. The study of extinct plants addresses these questions through the analysis of fossilized specimens retrieved from field studies, reconstituting the morphology of organisms that disappeared long ago. Paleobotanists trace the evolution of plants by following the modifications in plant morphology, shedding light on the connection between existing plants by identifying common ancestors that display the same traits. This field seeks to find transitional species that bridge gaps in the path to the development of modern organisms. Fossils are formed when organisms are trapped in sediments or environments where their shapes are preserved. Paleobotanists collect fossil specimens in the field and place them in the context of the geological sediments and other fossilized organisms surrounding them. The activity requires great care to preserve the integrity of the delicate fossils and the layers of rock in which they are found. One of the most exciting recent developments in paleobotany is the use of analytical chemistry and molecular biology to study fossils. Preservation of molecular structures requires an environment free of oxygen, since oxidation and degradation of material through the activity of microorganisms depend on its presence. One example of the use of analytical chemistry and molecular biology is the identification of oleanine, a compound that deters pests. Up to this point, oleanine appeared to be unique to flowering plants. However, it has now been recovered from sediments dating from the Permian, much earlier than the current dates given for the appearance of the first flowering plants. Paleobotanists can also study fossil DNA, which can yield a large amount of information. By analyzing and comparing the DNA sequences of extinct plants with those of living and related organisms. Through this analysis, evolutionary relationships can be built for plant lineages. Some paleobotanists are skeptical of the conclusions drawn from the analysis of molecular fossils. For example, the chemical materials of interest degrade rapidly when exposed to air during their initial isolation, as well as in further manipulations. There is always a high risk of contaminating the specimens with extraneous material, mostly from microorganisms. Nevertheless, as technology is refined, the analysis of DNA from fossilized plants will provide invaluable information on the evolution of plants and their adaptation to an ever-changing environment. The major divisions of land plants. The green algae and land plants are grouped together into a subphylum called the Streptophytina, and thus are called streptophytes. In a further division, land plants are classified into two major groups according to the absence or presence of vascular tissue, as detailed in Figure 5. Plants that lack vascular tissue, which is formed of specialized cells for the transport of water and nutrients, are referred to as non-vascular plants. Liverworts, mosses, and hornworts are seedless, non-vascular plants that likely appeared early in land plant evolution. Vascular plants developed a network of cells that conduct water and solutes. The first vascular plants appeared in the late Ordovician and were probably similar to lycophytes, which include club mosses, not to be confused with the mosses, and the pterophytes, ferns, horsetails, and whisk ferns. Lycophytes and pterophytes are referred to as seedless vascular plants because they do not produce seeds. The seed plants, or spermatophytes, form the largest group of all existing plants, and hence dominate the landscape. Seed plants include gymnosperms, most notably conifers, gymnosperms, which produce naked seeds, and the most successful of all plants, the flowering plants. Angiosperms. Angiosperms protect their seeds inside chambers at the center of a flower. The walls of the chamber later develop into a fruit. Section summary. Land plants acquired traits that made it possible to colonize land and survive out of the water. All land plants share the following characteristics. Alternation of generations. with the haploid plant called a gametophyte, and the diploid plant called a sporophyte. Protection of the embryo. Formation of haploid spores in a sporangium. Formation of gametes in a gametangium. And an apical meristem. Vascular tissues. Roots. Leaves. Cuticle cover. And a tough outer layer that protects the spores contributed to the adaptation of plants to dry land. Land plants appeared about 500 million years ago in the Ordovician period. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.